Hello. Today we are going to discuss histology of the normal kidney biopsy. We are going to discuss the normal structures that are relevant to understanding kidney biopsies done to evaluate medical disease of the kidney. What I mean by medical disease are diseases that are treated with medications as opposed to surgical disease, which are diseases that can be treated by removing something, such as a kidney tumor. Okay, let's get going. After watching this video, you should understand basic functions of the kidney. You should also understand basic anatomy of the kidney and nephron. You should understand how a kidney biopsy is obtained and how the biopsy tissue is processed into a glass slide. And of course, you should understand the histology of the normal kidney biopsy. The kidney is highly complex. One of the main functions is to filter blood and remove metabolic products and toxins by excreting them into the urine. It also maintains proper fluid balance, electrolyte balance, and pH in the blood. It also secretes renin, which regulates blood pressure. It secretes erythropoietin, which stimulates red blood cell production. It also converts vitamin D into its active form. This is a drawing to remind you of basic kidney anatomy. The structures you should know for today's video are the renal cortex, which is the outer portion of the kidney. The renal medulla is the inner portion of the kidney. The fibrous capsule is the connective tissue covering of the kidney. Here on the right, you have the renal pelvis, which is where the urine enters prior to entering the ureter. And of course, here is the renal artery and the renal vein. The nephron is the name we use for the combination of structures that carry out the functions of the kidney. Kidney physiology is highly complex, and we are only reviewing the names of the structures that compose the nephron. To give you context when compared to the previous drawing, the top beige region is the cortex and the lower orange area is the medulla. Here is the glomerulus, which is where the blood first gets filtered. This is the glomerular capsule, which has a visceral layer, parietal layer, and capsular space. Here on the right is the proximal convoluted tubule. Here is the distal convoluted tubule. Here is the loop of Henle. Here are the connecting tubules and the collecting duct. And here is the macula densa. Okay, now that we know some basics, let's listen to our nephrologist, Dr. Acott, explain how the core needle biopsy set works.
But I can show you. So when you prime it, you bring back your cutting needle first, your gauge needle second, and then when you click it, that goes forward, and then the cutting needle goes over it.
Here is our technician receiving the biopsy. You can see she is removing the kidney tissue from the needle with saline. Here we have our kidney biopsy in a Petri dish. Before we process the sample, we want to make sure we have good kidney to look at. What do I mean by good kidney? For most kidney biopsies, we want to make sure we have glomeruli. How do we do that? We can look at the biopsy under a dissecting scope. Here you can see the kidney biopsy as I move it under the microscope. These round structures are glomeruli. We'll talk a lot more about glomeruli later in the video. This looks like good tissue. Let's go ahead and gross it in. Here is our pathology assistant putting the kidney biopsy into a cassette and then into a metal tray containing formalin. Here, our medical laboratory assistant is putting the metal tray containing the cassette into a processor. The histology processor changes the solutions from formalin into alcohol, then xylene, then into paraffin. Paraffin is the same as wax. This process takes about 12 hours. Okay, the next morning, bright and early, our histotechnologist takes out the tray containing the cassette. Here she is putting the cassettes into the embedding station. Now she is going to embed the tissue into wax and put the cassette on top of the wax so she can keep track of the tissue. When the wax cools, she cuts thin slices of the tissue on a microtome and then floats the sections in a water bath. She then picks up the sections with a glass slide. Then she puts the slide in a machine that stains it with hematoxylin and eosin. Next is a machine that puts a cover slip on the slide. Here I have a glass slide of the kidney biopsy. Let's take a look at it under the microscope. Okay, in order that we can both see it, we need to do one more thing. Let's take a digital scan of the glass slide. The Aperio scanner equipment scans the glass slide with a microscope lens. It makes a digital image of the tissue. Now, we can finally look at the tissue together. Here is our biopsy. Let's start at the top. I am going to look at the piece on the right. Here, this pink fibrous tissue is the capsule. I am going to outline it with my pen. Let's continue looking at the rest of the biopsy. Here you can see 4 glomeruli. Let's take a closer look. I am going to outline 2 glomeruli. Let's take a closer look over here. You can see that these are tubules. Some of these tubules have large amounts of pink cytoplasm. Those are the proximal convoluted tubules. I will outline 1 proximal convoluted tubule with my pen. Some of the tubules have much less cytoplasm, such as this one right here. This is a distal convoluted tubule. I'm going to outline it with my pen. Now, I'm sure you remember at the beginning of the video, we named a lot of other types of tubules. In routine histology of the cortex, you can usually only see proximal and distal convoluted tubules. So those are the only 2 types I've outlined. Let's keep looking. Over here is an arteriole. I'm going to outline it with my pen in red. Okay, now we've outlined glomeruli, tubules, and arterioles. What else should we look at? The 4th major compartment is the interstitium. What is the interstitium? Interstitium is the word we use to describe the tissue in between all these other structures. In a normal kidney, you can't really see much interstitium. If you can imagine the small amounts of space in between the other structures, that would be the interstitium. Even though you can't really see it very well, I will outline some of it over here. Now, even though I only outlined a small portion here, the interstitium is present in between all the other structures in the entire biopsy. Okay, now we've looked at the 4 major parts of the kidney biopsy: the glomeruli, the tubules, the blood vessels, and the interstitium. These 4 major parts are very important for interpreting diagnostic specimens. Let's go ahead and take a closer look at a glomerulus. The glomerulus contains capillaries which are lined by endothelial cells. I'll outline 1 capillary with my pen. The glomerulus also contains podocytes. The glomerular basement membrane is in between the endothelial cells and podocytes. The 3rd major type of cell in the glomerulus is the mesangial cell. Now, in a normal kidney, it is difficult to distinguish these 3 types of cells just by looking at them. But in certain disease states, these types of cells become more visible. They can also be distinguished on electron microscopy. The white space surrounding the glomerulus is the capsular space. And this thin layer of cells surrounding the capsular space is Bowman's capsule. Okay. That's it for the structures that can be easily seen in a routine hematoxylin and eosin-stained slide of the kidney. After watching this video, you should understand basic functions of the kidney. You should also understand basic anatomy of the kidney and nephron. You should understand how a kidney biopsy is obtained and how the biopsy tissue is processed into a glass slide. And of course, you should understand the histology of the normal kidney biopsy.